So what you have in front of you is mostly right. There's some changes, and I'll point them out, and you're going to have to like scratch things out as you go. It's readable. I grant you that it's smaller print, smallish print, but it is readable if you get close to it, and it's a good source. It's a good example to have. On your exam on Wednesday, you can expect to see a question like this one, at least one, possibly two. And if you don't understand what's going on, you're not going to be ready for it. All right. Here's this corn that we've already done. You've already turned in the data from collecting it, right? Is that correct?
Yes.
You had the corn, you had the kernels, yes? You were absent. When you're absent, what should you be doing? You should be watching the video of that day's lesson.
I watched it.
Did you watch it? Then you did the lab, because I emailed you the lab as well. When I Jupiter grade message you stuff, and I send you videos of the class material, and you're absent, you had a whole weekend to get it ready, I expect you to spend some time going through that. So you got some work to do yet. And the exam is coming up, so get to moving on that. So as we look at this corn, I want to remind you that when you see a corn kernel or a grain of corn, just like a rice grain, a kernel grain, a corn grain or corn kernel is an embryo. It's an embryo, analogous to a baby. You can take any one of these and plant them, and they will turn into corn plants. They're individual corn plants. There are many different kinds of embryos here. Do you agree? Yeah. Each corn cob has 100 embryos, 100 babies. Each corn cob, there's many corn cobs per plant, so one corn plant can lead to many, 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 many embryos, many babies. Yes?
So each one of them is a baby.
Each one of these is like a human baby, right? An embryo. It's ready to go. Just plant it and it'll grow under the right conditions. Are we clear? So this is after fertilization, after the two haploids became diploid, right? Corn's a little more complicated, but we don't have to get into the genetics of corn. The bottom line is that this corn cob, these different colors are either dominant or they're recessive. The color and whether they're smooth or shrunken is dominant or recessive. By the way, you will have to upload these notes in Cornell Notes. So I see some of you not writing. I'm going to expect you to be, there will be an assignment. There will be expected of you. It'll be called classwork. There's no excuse for anyone not getting that done, right? So either purple smooth, you can write on that. And then you could also add some Cornell notes on the side, right? So you have purple smooth, you have purple wrinkled, you have yellow smooth and yellow wrinkled. And so you see that purple is dominant, right? I would write this down. Purple is on a separate sheet of paper. You have it there for your reference. It's there for you. You can highlight it in your notes. But I would write it down in my notes on a separate sheet of paper in Cornell Notes. That P is for purple, big P is purple, little p is yellow. So how do you get yellow?
Two little p's.
Thank you very much, excellent. Does everybody understand that? Mm-hmm. Do you understand that to get the recessive trait, you need two versions of it? If you have a big P and a little p, it's not yellow, it's purple. So smooth is big S, and little s is shrunken. So these are your four characteristics. Obviously, these are the recessive genes, and these are the dominant genes. In order to get the recessive trait, you have to have two copies of the recessive gene. The allele, right? The word allele. If you read this, you don't have to read the whole thing because we're kind of skimming through it, but you can read it at home if you'd like. We're gonna see that there's a table 1, we'll see it in just a second, that's a cross between the two heterozygote parents. Two heterozygote parents. They're telling you that. they're telling you that the two parents that are heterozygous have a bunch of embryos, and this is what we get. Now, these embryos ideally would be a corn cob. These are all the embryos from the two parents. Does that make sense? Mm-hmm. These are the products of this cross. Two heterozygotes, right? Big P, big S, little s crossed with big P, big S, little s gets you this. Okay? Does that make sense? All right. So do you have any questions? Because if you got a question now, it's just going to get worse as we go along. Yeah?
You said, what are you doing to like when you cross?
This corn cob. This is the results. So each of these embryos is a result of a cross between those two parents. So you get a new plant. That plant has all these embryos. That's it. That's the result of that. All right. Are we good? All right. Let's go on. When we do that cross, this is the ratio we would expect, right? So let me ask you to do this, because you need to be able to do this by Wednesday, right? Can you, taking two heterozygotes, which would be big P, big S, little s, crossed with big P, big S, little s. Those two parents, given those two parents, can you take these two individuals and come up with these embryos, or these, gametes? Do it. Do it right now on paper. Show it to me. When I ask you to upload the notes, I want to see you use foil. Use the arrows. Show me. Demonstrate you understand that these two parents, each one of these can produce these many embryos. I want you to do foil. If you don't know how to do foil, then the problem is, let me tell you where the issue is. When a student gets this on a test and they're asked to make something like this, what ends up happening is the student that doesn't know how to use foil will end up putting big P, big S, little s, big little p. They'll put all kinds of options here except for the 4 possible gametes, the 4 possible eggs and the 4 possible pollens, right? Does that make sense? Because what happens is kids don't understand how to create foil, how to create the embryos using foil.
You want us to write down the charts?
You can, but the chart is less important to me than how do you get this row and this column. How do you get the gametes? How do you get the gametes? These are the embryos, right? Does that make sense? These are diploid. They have 2 copies of each gene. These are the gametes. They only have 1 copy of each gene. So how do you get the gametes? Because the people that mess this up are the people that can't make the gametes correctly. They get confused. Why are students confused? Why does this confuse? It's not that you take this P and put it here, this P put it here, this S put it here, this S here. That's easy for people, okay? The hard part is getting this row and this column. They don't know how to make the gametes. How do you get the gametes in the right order? If you don't know how to do that, then you're lost. So I'm asking you to make the gametes right now. I'm going to do it with you in a second. I want you to try to do it on your own. And then compare what you did to what I do and see if you can do it right. Because you have to be able to do it right by Wednesday.
I don't understand what you're saying. I don't understand.
You don't understand that. You don't understand foil. Do you not remember foil?
No.
Do you have algebra right now?
Yeah.
With Miss Simpson or?
I don't understand algebra either.
Okay, but so you got to take it one step at a time. If you absolutely don't understand, then pay extra attention when I explain it in just about 30 seconds. But right now, what I'd like you to do is that you see here that there's 2 copies of each gene. I'd like you to come up with all the combinations of one of each of these that this creature can make, and do the same for this creature. How many different, remember you need 1 P and 1 S, how many different combinations can this creature make, and how many different combinations can this creature make? And I'll go through foil with you in just a minute. Now, the reason the algebra teacher taught you FOIL, and the reason I'm using the word FOIL, is because it's really the best way to get all the possible combinations in a very clear and concise way. So, FOIL, F.O.I.L. First, outer, inner, last. Right? All right. So, what's the first?
It's big P and big S.
Big P and big S. So, big P and big S. See? That's one possible gamete.
Big P and small.
That's right. And so now we go outer, so that's big P and small s. Perfect. Big P and small s. So that's another option. That's another possible gamete.
Little P and big S.
Very good. Then little p and big S, that's the inner.
Little P and small S.
Little p, oh, let me do that again. Little p and big S. And then little p and little s. The four possible gametes that this creature can make. What are the four possible combinations of genes that different combinations of genes?
It's the same thing for the other one.
It's exactly right. It would be the same thing for the other one because it's outer, first, which would be big P, big S, then big P, little s, then little p, big S, and then little p, little s. So you end up with the same thing is right, which is why these four are the same as these four. Because you have big P, big S, big P, little s, little p, big S, and little p, little s. These four and these four are the same because both parents are double heterozygotes. This part is where everybody messes up when you're doing a cross. When you're doing a dihybrid cross. Now, will they ask you to do a dihybrid cross on the end of course exam? I doubt it. But will they ask you to analyze one? Probably.
You said both parents are double what?
Heterozygote. I'm going to show you some examples I already have. We've gone through them last week, but I'll go through them again. Different examples where they're not heterozygote. And you'll see what the difference is between the two. Yes, sir. No, homo means the same, but there's no. How are these the same? Are you saying this one's homozygous to this one? No. Homozygous means the two copies of genes that you have are the same. Do these have two of the same P's? Do they have, are these two genetically identical for these two genes? Yes. Big P, little p, big S, little s. Yes. But homozygous means that your genes, the two copies that you have, are the same.
So like big P, small s.
Exactly. Big P, big S, small s. Those are homozygous. When you have two different individuals, right? They're not homozygous to one another. Homozygous means your chromosomes have the same versions of the alleles. Let's look at a homozygous, shall we? Let's take a look at what happens if one of these are homozygous for just one character. Big P, little p, big S. How many different gametes can this creature make? Do it. Do the FOIL. Do it on paper so you get the practice.
I like the Punisher back before.
Which one? No, what did you say?
I like the Punisher before.
The Punnett square. You can't get the Punnett square here of this dihybrid cross without FOIL. You have to use FOIL to get the possibilities because These are the 4 possibilities because you did FOIL. Now let's do FOIL here. How many different kinds of, only 2. 2, because look, big P can go with big S, but won't the big P with the big S be the same again? So there's only big P, big S for that first option. Now the second option is what? Little p, big S. Those are your only 2 possible gametes. Will you produce 4 eggs? Sure. But you'll either have this or this. You won't have 4 eggs with little p, little s. Why can't you get little p, little s?
Because
There's no little s. There's no little s. This is a homozygous dominant for S. This is homozygous dominant for S. Do you hear that? This creature is homozygous dominant for S. It's heterozygote for the B, for the P. Now what if you had a homozygous for both genes? What if you had big P, big S, big S? How many different kinds of creatures could you make then? Only 1. So understand, and please pay attention, because this is the FOIL again. If you did FOIL, big P goes with big S. If you did, no matter how many different, whether you did inner,
It's the same.
it's the same. It's all big P, big S. Only 1 option. Here you have 2 options. Here you have 4 options. Are we all clear on this?
Yes.
Because you're going to have to do this. All right, so given that all this is nice, but it's not, these are examples. This is what we had to do right here. So we had FOIL. We produced our 4 different gametes, the male and the female gametes, and they produced all these embryos, and the embryos are here, right? Let's go ahead and count how many different kinds we had. By the way, this is like the 3rd time we've looked at a 9 to 3 to 3 to 1 ratio. So you should be expecting a 9 to 3 to 3 to 1 ratio problem on your test, right? Okay. So it says 4 different phenotypes which define fractional ratios, 9 to 16 purple and smooth. Well, let's make those blue. Let's make those blue. 9/16 purple and smooth. This was purple and smooth. Why? Because you have a big P and a big S. All you need is 1 big P and 1 big S. Right? That's all you need to have purple and smooth. This is 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 purple and smooth. They don't have the same genotype, but this one's not purple and smooth. But the next one's what? Purple and what? Shrunken or shriveled, right? Why? Because there's 2 little S's. This is purple and smooth. That's not. This is purple and smooth. This is purple and smooth. This is purple and smooth. So how many is that? 1, 2, 3, 4, 5, 6, 7, 8, 9. 9. Right, that's good. But I just want to make sure you see it. That the pattern is the same. No matter what the genes are, the pattern's always going to be the same, assuming you have what? 2 heterozygote parents that give you these gametes. This combination is always going to end up the same, which is why we have you memorize 9 to 3 to 3 to 1. All right, so the next one is red. Let's see, it says purple and shrunken. So this is purple and shrunken, right? This is purple and shrunken. This is purple and shrunken. And those are the only ones that are purple and shrunken. Do you agree?
Yeah.
Okay. So there's 3 out of 16. Yellow and smooth. Yellow and smooth would be yellow and smooth, yellow and smooth, yellow and smooth. Again, 3 out of 16. And the last one is going to be yellow and shrunken, and yellow and shrunken, and we're going to color it pink. Yellow and shrunken then is just this last one here, this double homozygote recessive. Are we getting that? Because there was a question over here about what was homozygous, what is heterozygous. This is 2 homozygous genes here. This is homozygous for P, but heterozygote for S. These are both homozygous. This is homozygous dominant for both. This is homozygous recessive for both. Right? And let me tell you right now, I'm going to say it plainly. The key to every genetics problem, almost every genetic problem you're going to have, the very key, the foundation that's going to help you solve almost any genetic problem. Are you listening? Hello, Mr. I'm going to lay my head down and not pay attention. Tap him on the shoulder.
Charles.
Yo, sit up. Sit up. Why are you rolling your eyes at me? I don't understand. So, whatever. The bottom line is this: the key to solving all these problems is your homozygous individuals. Homozygous recessive is the key. If you have a homozygous recessive, you know exactly what it is. All you have to know, if you have a yellow and shrunken, you know what their genes are, right? If you have yellow shrunken, you know it's little p, little p, little s, little s. Guaranteed. Why? God bless you. That's right. You only get recessive with the recessive genes. You only get the recessive trait with the recessive genes. So if you have to have little p, little s, little s. However, if you have homozygous dominant, how do you know it's homozygous dominant and not heterozygote? You don't. Because you have, in both cases, you have a P. Look at this. Would you see a difference between these two? No, you would not. They're both purple and smooth. Right? Even though this is a heterozygote for smooth and this is a homozygous for smooth, you wouldn't know. So the key to all these problems is this homozygous recessive. Of course, if you did the genotypic ratio, this is what you get, but it doesn't help us too much when we discuss that. Yes.
I think you should give like a worksheet with like the bunch of them on there. And then we should like put either homozygous recessive or homozygous dominant or heterozygous.
Why don't we start first with doing this and making sure we understand each other and know what we're talking about. Taking notes, writing the notes down, studying our notes instead of asking for worksheets, right? Are you going to get worksheets? Sure. Are you ready for them? I don't know. I got people here that don't understand the difference. That just now, after a week of talking about this and doing problems, by the way, and doing a lab, still don't understand the difference between homozygote and heterozygote. Some people don't know FOIL, even though they covered it in algebra and here in this class.
You just said
it. Easy. We're not. No one's talking about anyone specific. This is about the fact: are you ready to do your worksheet on your own? I don't think so. Yeah. Question? Yes, you may. So what are the different number of phenotypes is 2 to the second power would equal 4. So you have 2 different genes, a P and an S, right? With 2 alleles each, you get 4 different phenotypes. What if you had 3 genes with 2 alleles each? You can get how many different gametes? 9 different gametes. So as you add genes with a dominant, with more than one allele, you start to see an exponential growth of how many different gametes you can have. Now take that number, put the number 10,000 there, and put 2, 3, 4 different, like for instance, blood type in humans has 4 different options, right? Can you imagine how many different gametes you could possibly have? And then you're recombining them, and then you're having sex, so you're having 2 individuals who make all those different gametes that can then recombine. That's why each human being is so individual. That's why you never have more than one person with your particular combination of genes, unless you have a twin, an identical twin. And even then, because of epigenetics and changes and impacts of the environment, your particular combination of genes is never the same. All right, let's take a look at a problem. This is the kind of problem you're going to have. Go ahead and write this out. Given which is dominant, which is recessive, go ahead and write it out. Make a little chart in your notes of what is given to you here. Remember that big P is purple, little p is yellow, big S is smooth, and little s is shrunken. Go ahead and write it out. I'll pause. pause here and then we'll come back when I've given you a couple minutes. This is your practice. You wanted a worksheet? This is your practice right now. Do it. Write down in a chart. This information is giving you a phenotype. Write out what it is, the genotype, given this phenotype. Go. Okay, so let's go ahead and do this. Now that some people have tried and others have wasted time, let's go ahead and do this together. So we have 381 grains total. Now what does that mean, total grains? Remember, what that means is that there's 381 corn kernels that we counted on a cob, right? Does that make sense? 381 of them. 381 embryos,
Yes.
right? 381, not gametes, embryos. All right.
You divide that by 2.
Of those 216, let's go ahead and write it down, 216 are purple and smooth. Purple and smooth. So what's the genotype for purple and smooth? Big P, and the other one can be either little or big, couldn't it? So either you can do it right at one of two ways. You can do big P, little p, or big P, big P. I think that's a waste of space, and it gets confusing after a while. So what I would suggest is you write big P blank, or big P question mark, whatever you want, because it could be either one. But we know there's at least one big P because it's purple, right?
Yeah.
All right, and what's the S? Smooth, so that's what? Big S blank.
Yes.
All right, so it could be, again, either big S, or big S, little s. We don't know. This is why your recessive is so important. So that's this one. (216) purple and smooth. The next is 79 purple and shrunken. So now 79 are purple and shrunken. So purple's what? Big P, blank, and then shrunken? Little s, little s. There's that homozygous recessive. It's always so important because it just tells you exactly what the genotype is. There's no question it's little s, little s. How do I know that? Because it's shrunken, and shrunken's recessive. The next 65 yellow and smooth. 65 yellow and smooth. Yellow is what? Little p, and smooth is what? Big S blank. We're not sure what that is. It could be another big S, it could be a little s, we're not sure. And our favorite child that gives us all the information in the world is this 21, which is what? Little p, little s, little s. We know these 21, we know exactly what their genotype is. That's what's so wonderful about these homozygous recessives. And you'll see when we do a test cross how important they are. They give us all the information in the world. They allow us to solve all kinds of complicated genetic problems. All right. Sorry about that. All right, so your hypothesis is that you have this ear corn, ear meaning the corn cob, and it's produced by a dihybrid cross. Your hypothesis is that this cross was between two heterozygotes. So two pairs of heterozygote genes, resulting in a theoretical expected ratio of what? 9 to 3 to 3 to 1. Right? You saw the table, some people wanted to make it, so that's what we expect. Well, how do you know? How do you know? This doesn't say 9 to 3 to 3 to 1, does it? It says 216, 79, 65, and 21. That's not 9 to 3 to 3 to 1. Say that again? How are we going to get this? So everybody wrote this down, because if you don't have this written down, when we go to the next page, you're not going to be able to discuss it. So make sure you have that. It's this part right here that you need, right? All right. So let's move on. This is the this is the graph that this is what you're going to have to be able to do chi-square. How do we know that this data, that these results actually prove that this stuff, that these genes are in fact the result, that these results are the result of a cross between two heterozygotes? How do we know that this is a dominant recessive inheritance at all? How do we know that there's not some other kind of genetics happening that we haven't discussed yet or discovered yet? Well, we're never for sure, you're right, but there is some evidence in chi-square. Chi-square is used everywhere, by the way. It's used in business, it's used in law, it's used in obviously in any kind of statistics. It's a statistical analysis that lets you know how closely do your observed results, how close are they to your expected results? That's what chi-square is. So to remind you what chi-square is, let me write down the equation. You wrote it down last week. Had you done your job in Cornell Notes, you would have written it down and studied it, and you would be able to tell it to me right now. What is chi-square? Chi-square is what? What's the formula? You spell it chi-square. What is chi-square? What does it equal? It's the sum of the observed minus the expected squared divided by the expected. It's the observed, what you actually see when you do the experiment. What is it that you see? Or when you count how many car crashes are teenagers involved with, how many did you expect there to be? And is there a difference? Is there something to the idea that teenagers get into more car accidents than older drivers? So we can do what we do when we have that question, any kind of question, is we collect data. We count how many accidents are there. We count how many accidents do teenagers get involved with. And we look at how many we expected. And we do this calculation, and it tells us if the number that we expected is really far away from what we observe, then maybe our theory's wrong. There's something wrong with our hypothesis. This is testing your hypothesis. It's figuring out how close are your actual results to what you see. So obviously it makes sense that we involve both the observed and the expected. So how close are your observed to your expected? Well, you subtract the two, and that tells you the difference between the two, right? And the reason we square it is so that we get no negative numbers. And we divide it by our expected, so that gives us a number that's like an average. Like let's just say it gives us some kind of relationship, a ratio, right? That ratio then, we can add them all up and come up with chi-square. That's what this symbol means, the sum of. That's what this means, the sum of. Anytime you see this in calculus, if you see it in algebra trig, if you see it anywhere you see it in life, in the statistics, if you see this E in math, it means the sum of. We add a bunch of these together. We're going to do this a bunch of times, and we're going to add them together. We're going to do this, in fact, once for every one of our phenotypes. Does that make sense? So then we're going to add those together. So we have our purple smooth. You have this already written down, so you don't have to copy it. Yes, you may. You have purple smooth. You have 216. You observe 216 purple smooth. 216. Now, the way they do the observed ratio is they take. By the way, this is yellow shrunken. Do you see this? This actually belongs up here, okay? This was just cut off on the page. So the way they do this, and you can do it more than one way. You could take, the observed is what you see, right? We already wrote those numbers down. Do you agree? That's where these numbers, 216, 79, 65, 21, we literally just wrote those numbers down here, 216, 79, 65, 21. That's where we got those numbers. Is that clear? We observed them. We counted them on the corn cob. The observed ratio is how many to what? Now you could do a ratio of 216 to the total, which would be 381, or you could do 216 to 21. Either way, it's 216 to 79. Why do we do that? Because one way of doing that, one reason to do that, is we know that 21 should be the number 1, right? Because it's 9 to 3 to 3 to 1, right? Do you agree? So we could say, what is this compared to this 1? So if we take 21 and divide it by 21, what do we get? 1. We take 65 divided by 21, we get what? 3.1. 79 divided by 21, we get 3.8. 216 divided by 21 is 10.3. Because it's 9 to 3 to 3 to 1, right? It simplifies the math. Instead of showing 9 to 16, 216 to 381, what is the ratio, simplifying it, dividing it? This simplifies things. It makes things go quicker, yes? So that's an interesting way of doing it. I had never seen it done that way, but I'm not a statistician, so maybe this is common. So these are your observed numbers. This is your observed ratio. This is what we expect. We expect 9 to 3 to 3 to 1, right? Does that make sense where we get our expected? All right. Please make sure it does, because when we do a regular cross, we expect 3 to 1, for instance. When we do this number, these expected might change depending on the problem you're dealing with. And we'll talk about that later. So know that the expected ratio is, because you know this is a heterozygous cross, you expect to see 9 to 3 to 3 to 1 in the genotype, in the phenotype of the embryos. What do we get? 10.3, 3.8, 3.1, and 1. Are these two close enough?
Yeah.
Is 9 close enough to 10.3? Is 3 close enough to 3.8? Is 3 close enough to 3.1? I don't know. You say yeah. Why do you say yeah? How do you know that means that difference doesn't mean doesn't equate to something being wrong? That this is not dominant recessive. That these are linked or these are not linked. Huh? The answer is you don't know. Yeah, I'm glad. You don't know. And so that's why we do this whole chi-square thing. The reason we're doing all this is to try to figure out, is this close enough or not? When you're done calculating chi-square, you're going to compare it to a table. And that table, that comparison is going to tell you, this is close enough or it's not close enough. If it's not close enough, you have to reject the null hypothesis, and if it's close enough, then you accept the null hypothesis. And we'll talk about what the null hypothesis is in just a second. All right? So, you take your expected, what's your expected number if your expected ratio is 9? It's 9/16 times 381. Where'd you get 381? The total number. There's 214, that's 71. So 71 compared to 79, 71 compared to 65, 24 compared to 21, and 214 compared to 216. That's what you're expecting. Once you have your expected number, you have your observed number, yes? Now you can do this. Observed minus expected squared divided by expected. Where'd I get 4? 4 divided by 214 equals this. Where'd I get 4? Do the math. I see, I hear you. You're right. I can think you're right. If you're paying attention, you should know the answer. Should I make this a quiz? Have you write it down on a piece of paper, turn it in? It's observed minus expected. What's observed? 216 minus expected, which is what? 214, which is what? 216 minus 214 is what? 2. But this is 4. What? Squared. It's this minus this squared. Observed minus expected squared. Divided by expected, so 4 divided by 214 equals this. Let's do the next one together. Understand you're going to have to do this on the exam by yourself. This is subtracting and adding and squaring using a calculator. I don't see this being a problem, assuming you understand what's going on here. Observed minus expected squared divided by expected. Let's do it here. Where did they get 64?
And they squared it.
Observed minus expected. How many did we expect? Which is how? What's the difference? 8. 8 squared is divided by 71 is this. It's pretty straightforward. Now the next one is 36. Where'd they get 36? 65 minus 71. That's a negative number, but when we square it, it's not negative anymore, right? What's 65 minus 71? 71 minus 65. 6. It would be negative 6 if it's 65 minus 71. Yes, it's negative 6. Negative 6 squared is 36, positive 36.
Oh, that's
That's why we square it, so we have no negatives involved in this calculation. Divided by 71, because that's expected. It gives you this number. See, the closer this is to 1, the closer that you are to your expected. If you get a 1 here, if this is a 1, you're very close to your expected. You want to get, it would be very spot on if it was all 1. It almost never happens in real science. Yeah.
So all you gotta do is find the square root of each, like the square root.
The square, not the square root. Just you're squaring
I'm
it.
talking about for the observation number, like 4, 64, 30.
If you took the square root of this, what would you get? The square root of 0.05, 0.507?
No, for like 4, 64, 36. To find your difference between your observation number and your expected number.
All you do is take your observed minus your expected.
I know what I'm saying. Like, you could just do the square root to find it out.
Square root of what?
Like, yo, what's that? I don't know if that's the observation number.
You mean this number?
No.
Take the square root of this?
Like 4, 64, them numbers, 36.
What do you see? What do you see 464? Oh, these here?
Yeah.
Oh, yeah. Yeah, but you won't know that. You don't know what this is. This is the square, taking a square root of this would just give you the difference between the two.
That's what I said.
So it would be 2. So that would tell you the difference between the two. If you took the square root of this number, this value right here will give you that there's a 2 difference, but you don't know what the observed and the expected would be. You have to have this filled in. It doesn't do you a lot of good to go backwards in this problem. Yeah.
When you get this, where do you get the 16 from? Is that
16 where? Where do you see the 16?
So it's like 9.
Oh, this 16?
Do you just add all of those, like 9 and 33?
9, 3 plus 1 is 16, that's right. And remember, if you did the dihybrid cross, remember that, look at the chart that you have, that I published for you, that I printed for you. If you count the total number of embryos in that dihybrid cross, how many do you think there are?
Wait, which one?
The dihybrid cross.
Oh.
There's 16.
This right here?
I can't see, so you have to bring it around here. Quickly. But if you look at the dihybrid cross, look at how many embryos there are. Remember we highlighted in blue and yellow and that? Yeah, that's it. Count how many you have. 4 by 4, it's 16. 16 different embryos, yeah?
Okay, so in the expected number column 5, how did they get equals 214 and equals 71?
Okay, so if you take 381, do you have a calculator in front of you? Get a calculator. Use your phone. Do the math right here now. right, so let's do this. 381 times 9, do the math. 381 times 9, and divide that by 16. Tell me what number you get.
Divide by 16 or
Yeah.
9
Well, it's multiplied. See, 381 times 9
over 16.
divided by 16.
Okay, I did that. Okay.
And what'd you get?
214.
Well, 214, yeah? Let's do the next one. 381 times 3. Do that. Divided by 16. What'd you get?
Bless you.
71, yeah? So all it is this number, which is the total, times the fraction that you expect it to be.
Are these gonna be on the test?
What?
Like these problems? Like, are you gonna give us problems?
I literally said that 3 times.
Okay.
You are gonna get a problem like this on the test, yes.
Are we gonna get calculators?
You need to have your own calculator as part of your supply list. Do I have some extras? Maybe. I might have some laying about, but I don't have enough for everybody. You need that. No, you may not use your phone on the test. Are we clear on this? How do we get this?
Yes.
So all you're doing is just taking your total, multiplying times your expected fraction, that gives you your expected number. Then this is just simply a squaring and a subtracting. All right. Let's go on. When chi-square is the sum of all of these, so all of these, 0.019, 0.901, 0.507, 0.375, added together, give you chi-square. Your chi-square value is 1.8. That is chi-square. You figured it out. Once you have summed it all up, you have your chi-square value. But that by itself doesn't give you any information, does it? I got 1.8, yeah! Well, that doesn't tell you anything. You need to compare it to another number. Are you ready? But this is the whole point. This is your chi-square. This value here, chi-square, is a sum, and that's what this means here, sum, is the sum of this all added together. So all these added together give you your chi-square. Is that clear? All right. So let's go on and see what we got. So 1 point step then. So this is chi-square. Oh, I missed something. Our chi-square value. So let me write that down. So your chi-square value is 1.8. Now let's see what we have, something that we have to calculate, and that's our degrees of freedom. You have to calculate your degrees of freedom. This is the simplest thing you'd ever have to do in any math or science class. Your degrees of freedom are the total number of variables, total number of options you have, minus 1. How many different options do we have with these two parents? How many different options do we have? Why do we have 4? You're right, it is 4. What are those 4? Purple and smooth, yellow and smooth, purple and shrunken, yellow and shrunken. Those are our 4 possibilities. The degrees of freedom is your possibilities, which is 4 minus 1. So in this problem, it's 4 minus 1, which is what? 3. So your degrees of freedom are 3. Your degrees of freedom is, in this case, 3. That's what you need. Now, we're going to look at this table. Now that we're going to have to figure out what this probability is. Now that we have our degrees of freedom, now we have to figure out what this is. Now, let's be clear on what this means. In this class, and in most of the time, you're going to be looking at the 0.05 probability. Yeah, you're going to have this big table. In some In some circumstances, somebody might tell you, I want you to use 0.95, somebody might tell you want 0.2, somebody might tell you want 0.1. But in this class, and in most classes you take, you're going to assume, I'm sorry, not 0.5. You're going to assume at 0.05, 0.05. You're going to look at 0.05. Is that clear?
So yeah, I heard my.
Okay, that's all right, that's all right. So it's 0.05 is what's significant or not. Do you see where it says it's significant? So really, this is where we want to stick to. At a 0.05, if there's a difference, so what does this mean at 0.05? What does this chi-square mean, and what do we do? We're going to have to wait out for that tomorrow, okay? We're going to talk about that tomorrow. So read the rest of this tonight. It's going to be a little complicated, but I think you'll see it'll make sense tomorrow. All right?